to invite Dr. Kwon, who comes with a delegation from Korea. Dr. Kwon is a principal research scientist at the National Institute of Agricultural Science. He is a part of the Rural Development Administration in Korea. But it's a long biography, which you can have a detailed, a rich one, and simply highlight a few things. So, master and bachelor in agronomy from the National University of Korea, a PhD from Coventry University, the UK, in plant stress physiology. And then Dr. Kwon has taken part of a lot of sort of government initiatives. I like a few of them. I mean, in part of the international negotiation, including the Convention on Biological Diversity and Biosafety Protocol. He's been involved a lot also in sort of R&D initiatives in the Korean government in relation to farm industry coalitions as well. You are part of a future strategic team on from the Korean Ministry of Food, Agriculture and Aquaculture on forestry to formulate the green growth policy in agriculture. That's still ongoing, I guess. And then, okay, these are only the attribution that he had with the Korean government, but he's got also a long career as a scientist, working with the National Institute of Ag and Biotechnology in Korea, and with a few short stints at the US Salinity Laboratory in the US, and you've got also a 2-year stint with the Biodiversity International, so he's also familiar with the CG family. So I think that's all I have to say to introduce Dr. Kwon. The floor is yours.
Thank you so much, Vincent. That nice introductions, long introductions. I am working for the National Institute of Agricultural Science, and the central government body working for the Korean agricultural R&D. We are over Korean agricultural R&D. So that I'm not quite speak fluently in English, but that's crazy. I am Korean. I can talk in Korean quite well. Anyway, that I am very happy to be here today. I also that I have to say big thanks to Dr. Sharma, and also that Vincent hosting me and showing me some fantastic achievement here. Also that project also yesterday I went there. Yeah, that I got some good idea for the future collaboration. Thank you. Before starting, maybe I would like to share the Korean drought in the Korea. But my title is actually the title for the presentation. It is Phenotyping of salt forest using physiological traits in cropland. Yeah, that you may already ask me why salinity in Korea, why drought in Korea. So that I'd like to give you an answer through my presentation. Okay, that every time history is good lessons for the future works. Here the ritual ceremony here, actually, ritual ceremony for rain. In Korea also got a drought. And you may be curious of how Korea belong to the temperature zone, suffer drought. Yeah, this has a long history of drought, especially in spring season. Korea has that almost more than 1,000 millimeter precipitation per year. Even that we suffer regular drought, especially in spring and the winter season. You know, how many people have we been to Korea? Is there anyone? Okay. Then it has 4 distinctive seasons, and all rainfalls come, majority actually rainfall come in the summer seasons, not the spring and winter seasons. So that when farmers farmers have to wait for the seeding, especially in the spring seasons, especially the rice production. With drought in the spring season was a big problem. Actually, here you can see ritual ceremonies. Still, sometimes some village areas they do ritual ceremony for seeking rains. Is it effective or not? But as a scientist view that we have to develop drought tolerant cultivars, especially for spring seasons. That is my interpretation. Or official record, last 500 years ago, 500 years, Ejo dynasty ruled Korean peninsula. That the dynasty actually recorded officially the drought each year. That we could see we also I myself cannot interpret this as what it means, but I can see some word here that indicating drought. It happened regularly last 500 years. But 1500 years ago, Korea, some the time the Korean government regimes were collapsed regularly because of the drought. And when drought happened, they have to change the kings because that is the way to sort out drought problems, not now. And the rain measurement actually that 1442 that we set up about the hydrology for to relieve the drought. So that when you visit Korea, that I strongly recommend to visit Cheonggae River. That in the center of the Seoul city, that is a big, nice actually Cheonggae water streams here. That they put the hydrology methods here to control the waters. At that time, no waters, no agricultural production. So the king ordered to invent this sort of measures to tackle the drought, especially in the spring seasons. Can you recognize her here? Can you recognize it happened last year? Irrigations in the spring season after they did rice transplanting of rice seedling, no water for 2 weeks. So the Korean president came to the agricultural field to irrigate this rice field. Yeah. It happened last year, the drought. So the drought here also, we can show them what is the actual drought for the agricultural production. That is what I like to share with you, Korean drought in the Korean peninsula. That this is not just the introduction. I like to talk about the salinity problems in Korea. As you see, maybe this one unique picture of the Korean agricultural system. This one protected plastic film cultures. As I mentioned earlier, that we have a 4 distinct seasons, especially in the winter season. We cannot grow any vegetables, but the protected plastic films actually good resources to produce fresh vegetables year-round, even in the winter season. Sometimes we warm up within this house structures. Actually, this one well developed for Korean agriculture. It covers almost 60% of Korean agricultural GDP productive. That's so that it's good meaning of economics. But recently we found some challenges. High salinity accumulation within the plastic film houses because of the even we irrigated well, but sometimes the dry conditions cause the salt accumulation. So you can see here that some damage due to the high salinity in the production of leafy vegetables. Also the strawberry. Maybe you are not familiar with the strawberry, but this is a good season for strawberry production. But it happened. The plant damage due to the high salinity. So that also the rice seedlings sometimes after we removed this plastic film houses, that when we planted rice seedlings, we got some high salinity caused this sort of problems. This one actually the Korean our challenge to tackle in the future as well. So you are not much familiar with this reclaimed high tidal saline land, especially coastal area. The Korean peninsula is quite small. That unluckily I cannot put here North Korea because I have no information on North Korea. So that here in the coastal area with reclaimed high tidal soils, you can guess quite enough salinity to kill all crop plants. So that we invented some good hydrology, but that's not enough. We have to invent sort of tolerant crops. One example here recently that we put up the big bank here to isolate this coastal land from the sea areas, that 33 kilometers. Good driving course actually. But here is that Korean government invested huge grant to expand agricultural land here. That actually we have needed sort of tolerant crops for here. That is 40,000 hectares. But maybe here is huge land, but in Korea that's small land. So 40,000 hectares quite big ones. Also, sometimes land price is very high in Korea, high density of population. So this new agricultural lands, especially at highland area, high tidal land areas, needs very specific crop plant, special sort of tolerant crops. Actually, total area covers all over reclaimed tidal land, saline land is about 350 hectares, which is big ones. So in terms of R&D, that we needed to tackle certain soil tolerant crop plants against ranging from 4 to 30 Siemens per maybe we cannot do this high salinity, but we have to target 4 to 8 Siemens. That could be a good target for agriculture product. So my job actually is plant physiology. Plant physiology Vincent. Yesterday and this morning I went to the Regis camp, phenotyping facility. So I visited here with my colleagues and how each tactic of phenotyping in terms of physiological trait. So that it's a fantastic phenotyping facility. But maybe Vincent may agree some points about why we have to do assessment of physiological trait as, for the sort of tolerance, especially for phenotyping terms. My colleagues, almost 50 scientists within my department have a specialism on molecular biology, not the plant physiology. They explore lots of genes, huge numbers of genes, and they like to insist I have a salt tolerant genes. Then actually I look around how have you done so far for the salt tolerance assessment. It's sometimes quite ridiculous. They got their own very regional ways. They put that as their own transgenic plant, the salt with the salt tolerant genes in the test tubes. Very less transpiration. They say they put the 400 millimole sodium chloride as a treatment, then say that the survival, okay, it's a sort of tolerance. But I asked them, do you have any idea? Can you use your plant for the farmers in high tidal land? They say, no, I have no idea. because that he just concerns about the patent and the publications. Maybe there are good approaches for publication and intellectual property right. But for farmers' views, that is not sort of because survival, not just any evidence of the productivity. So that generally that my colleagues concerns about genes, targeting on genes, and they impose very high salinity, sometimes 200 millimoles to 400 millimoles salinity. 400 millimoles almost equal to this sea salinity. We cannot grow any crop plant with the seawater. If we do that, maybe we can get the Nobel Prize. That we cannot create such crops. And also that they impose very shortly the stress. That is not realistic for field conditions. I like the Regis because this one field phenotype. But that is not also that their selection criteria quite different from the breeders or farmers criteria. Yes, they use the indication of survival or not. Then they publish the papers. Okay. Then sure they do the experiment in the laboratory, not in the fields. So, but breeders and farmers are quite keen on the yields with the various strength lengths in the field conditions for the salinity, even that the coastal reclaimed source, various salinities. Majority of the productivity of crop plant in the produced at the less or the moderate salinity. So that farmers need some long-term salinity treatment, not just short-term, because they have to grow whole life cycle of their own crops. So that I believe that physiological trait can narrow the gaps between gene works and the plant breeding for salt tolerance. So I set up those many low throughput phenotyping protocols for salt tolerance, especially in rice crops, rice plant. Then especially that you may know already that salt tolerance can be achieved by the multiple physiological traits, not just one or two single traits. That is not easy actually. So that we set up all some physiological traits here that especially the water relation measurement that we can measure the active osmotic adjustment, water use efficiency. For the ion relations, we set up the ion selective uptake on the transport, also compartmentation or sometimes exclusions. We like to assess those traits. And then maybe secondary damage by this salt stress can be the oxidative damage. That is that so far we cannot do this one. But all the physiological parameters can indicate the certain function of the genes. So that I put that one dishes, Korean traditional dishes, actually we call it bibimbap. That means that we have to prepare each element to achieve best taste. So that each physiological trait is each element dishes here. So that we put that very precise assessment of physiological trait for salt tolerance. So another concept here that actually we set up a pipeline to develop a salt tolerant transgenic plants. Actually, many of my colleagues working for the molecular biology actually concerns in this part. They do not concern much here. So that I like to build another steps here, evaluation and selection at all plant physiology levels here. And especially that we consider about the environment and the genetics to select the best type of physiological trait for certain target genes. Actually, then we have to explain how the genes can maintain the plant growth under the salt stress conditions. Okay, another point actually that what I got the concept at this moment, that the absolute two terms tolerance, absolute tolerance and the relative tolerance. You can see here that A and B could maintain the growth against the severity of abiotic stress. Even high, they can maintain growth. But like C types, at the control conditions, very high absolute plant growth. But when we impose the stress condition, it almost reduced very much the ability of growth because of it has no functions of tolerance, tissue tolerance. So that maybe that B types, B ecotypes, I don't like B ecotypes because it cannot have enough productivity in the very various salinity conditions. So I like the A type ecotypes for salt tolerance. Whenever I assess transgenic plant for salt tolerance, I try to find A types. So let me show you. So that I like to introduce three genes, what we explored so far, and actually I tested more than 20 genes, but this one, three genes show the certain physiological trait explaining certain maintenance of growth. So the one gene she repeated red binding factors, and also KEX2 genes that are also CIPK genes. That actually that we put that any sort of crop plant that for the rice. My department has a very reliable protocol for gene transformation for the Japanese rice and also potato and a few more crops. So we inserted these packagings to each crop species. As you see here that CVF genes that we invented that CVF transgenic potato here. We assess physiological trait using a water culture system, within the water culture system. You can see here that actually the general ones, when we impose stress planted, salinity stress that planted expresses CVF grape genes, and then actually help the cell growth, as you know, that we can expect growth maintenance because of these genes. And then actually, whenever we insert the genes, we do the very fine molecular characterization, including northern blot analysis. Then we confirm the all gene expressions. Then actually, that when we assess the whole plant physiology in the water culture systems, we found that actually we imposed 50 millimole sodium chloride for 15 days. Then actually that we measured the growth and the physiological parameters. Actually, you see in the photos here, actually CVF genes and the wild type plants can grow very equally at sodium chloride free conditions that we could not distinguish the growth. But in the presence of salinity conditions, CVF genes, CVF transgenic potato could maintain 20% more growth, each that we repeated more than 10 times. It happened. What happened? Why these transgenic plants can maintain better growth maintenance? But they mentioned their cell growth, so that at all plant level, not clear evidence. So that this one's, but another what we found actually this transgenic plant has an ability of growth maintenance of the salt stress with the doubled osmotic adjustment in the expressed cell shapes. What I found actually that something cell expansions, and plus that the osmotic, active osmotic adjustment. But I could not see such a big difference in ion accumulations. That is usually the potassium versus sodium ratios. There is not much difference, but big osmotic adjustment made by the CVF genes. That is what I found so far. That actually, that is a physiological phenotyping. So that we try to explain CVF could achieve less growth reduction due to greater osmotic homeostasis. That is what I try to explain. Another point, actually, the KSIConnect genes. Actually, that this one is quite well known for the ion uptake, ion movement of the plasma membranes. Actually, here, vectors and cytosol. Actually, God, you may not familiar with that. Actually, in Korea, this time here, actually, the watermelon seasons. Korea also produce watermelon, especially with the grafting systems. We needed a stock, reliable stocks for salt tolerance. That actually this is a growing market. The stock seeds sometimes God seeds market almost reached to $6 million, almost $6 million. About. Actually, that KSIConnect genes we isolated from Arabidopsis and transformed into garden plant, and that is actually the garden plant used as a grafting stock of watermelon. Then actually we needed a salt tolerance in stock plant. So that the transgenic and we tested the transgenic and the wild type garden plant and the aerated water culture system that within the growth chamber, surely. But I cannot control. Maybe environmental interaction could cause big troubles to interpret data. So that within the growth chambers, actually, surely we did that molecular characterization before doing these jobs, and about the 30 millimole salinity was imposed for 10 days. That slightly reduced because this plant is not much strong for the salinity tolerance. So then actually we found here the difference between transgenic plant and the non-transgenic plant. Actually, there is that surely the transgenic plant could maintain quite well in the presence of salinity compared with the wild type. But that actually the transgenic plant show the great biomass and the leaf area, and then we try to explain why actually the transgenic plant showed the great potassium concentration in shell shafts, and also the less osmotic potential in the shell shafts. That is actually more potassium uptake in the shell shafts could help to maintain this growth maintenance. That actually we like to do such a phenotype. Another one actually, the CIPK genes in rice. Actually, you already know that CIPK genes that we found that antiphoto genes. There is the CIPK to quite help this antiphoto genes. Then the CIPK transgenic plant is maintained great biomass under such stress conditions, with the less sodium and the greater potassium sodium ratios. As you see here, overexpression of the CIPK helped very much the more biomass accumulations as compared with the null or knockout lines. Also, the much less sodium content in the shell shaft compared with the knockout line. Here, also, so it could maintain well ion homeostasis as a tolerance mechanism. So CIPK could achieve salt tolerance through this mechanism for plant levels. So that is what I have done with the three genes, just the introductions. Maybe I hope we can bring such genes to phenotyping in the field using that Vincent system. The system is wonderful for the field conditions. Another one actually that you know that we invented the simultaneous measurement of water take and transpiration. With rice, maybe not, we cannot order crops easily, but focalism, you know, you are much know better than I do for the focal. Yeah, that actually another one actually that I compared the two genotypes to test my idea. Long-term salinity effect on water take and transpiration. As you see, actually, that when we impose it, it is a very general knowledge that salinity will cause the reduction of water take and transpiration. But I tried to measure simultaneously to the two parameters using my own system. I put that relative the transpiration and water take as expressed gram per minute per 100 gram pressure Celsius, or against the times per days. I measured many days here. Actually, that inverse the triangle indicated that actually water take that round mark represent the transpiration. So that red lines indicate the salt stressed one. Green lines indicate the plant grown in optimal conditions. When we measure here, that we found the salinity stress, salt stress caused very less gaps of the red lines here, less the gaps of the so the difference between water take and the transpiration compared with these green lines. Here is the gaps, but here actually in the daytime, even here the rice plant actually transpire a lot, water take a lot, and so but here and so surely that in daytime that water take and transpiration goes up, even salt stress plant. That my idea actually, what is the difference between water take and the transpiration when we measure it simultaneously? That is that I try to explain with that focally and IR29. IR29 quite well known for salt, sensitive cultivars, well known breeders, not my, but focally is salt tolerant. I noticed here that the difference between I actually defined water flux here, just the difference between water take and transpiration. I saw that here in the daytime, much fluctuated. Maybe my system is not working, not much accurate to reduce the outliers. But here, but in the night time, as you see, it's quite stable for transpiration. Just even stress and closure is not less concept. So the water take in the night time quite important for focally to achieve salt tolerance. Here actually focally could maintain much water, higher water take compared with the IR29. So this is what I found. So the conclusions, physiological phenotyping using physiological trait is quite useful to understand the function of the genes at all plant levels. That is what I learned so far. So but this is not information for the field phenotyping. That is not this information, just fundamentally understanding the function of genes. This information can help us, but still we need to do certain works for phenotyping in the field conditions, interacting with the environment. This one's actually within the growth chambers. Okay. Then actually the physiological trait, phenotyping using physiological trait is useful for also crop breeding. So that I have a certain client they do not pay me any single penny, but they say that they are my client. I like to provide this R&D service to them. They say that not payment, but anyway, I am happy to help them. Sometimes they bring the transgenic plant or the genes to my labs, so that to do this sort of jobs. Anyway, thank you so much for your attention, and thank you so much again for allowing me this talk. Thank you.
Can you tell us a bit more detail on Kokani and IR64?
Mm-hmm.
You measure the night transpiration.
Mm-hmm.
But why that would lead to would you infer that there would be sort of water accumulation at night?
When I did this one, actually I liked the water tank. That is the root has certain functions for salt tolerance, especially in nighttime. You may read some papers actually. Water take ability in the nighttime quite important. When the transpiration is quite low, focally has certain root functions to take more waters. Maybe that morphologically or some, I think that I do not know exactly why it has more water take in the night time, but it showed that this data significantly showed a big difference focally and between focally and IR29 water take ability, especially in the night time. That is what I found. Is it because of some hormonal signaling, the morphologicals, that we need to investigate?
Yeah, I said comment to that. You mentioned in your that molecular biologists they are more worried about papers, publication, and IP.
Mm-hmm.
But both in you showed us CBF and that's true.
Mm-hmm.
So both of them are independent.
Yeah.
Why did they decide on what you do?
Okay. That is good question. That our first genes actually CBF that we isolated from different crops. There are different genes. So what is isolated patently in the other parts. So, but we also the CIPK that is our own original DNA. I mean that we actually invented the genes. CIP, the KEX genes that we actually have. Our partner in the United States provided the gene to us. So that is a patent in the USA, but with patent for the transgenic crops. Is it okay?
So we'll leave it here. We'll give a round of applause for Dr. Kwon once again. Thank you very much.
Thank you.